Hey guys, how you all doing? I really hope that you all are doing good and the microscope that we have spoke about so far are crystal clear in your head. But if still not clear, I am saying to you again and again, please shoot your doubts in your comment section or we can just take an extra class comprising all your doubts. I hope that's fine. Okay, so now let's talk about confocal microscope. And as a ritual, let's see how the confocal microscope looks like. Now, this microscope, of course, looks different than any other microscope. The reason is here we are not using visible light rays. Here we're going to use UV light rays. And as we all know, UV light rays are harmful to the human skin and they can damage our eyes and they can be very, very dangerous to our DNA as well. So we are taking all necessary precautions to ensure that UV light rays only hit our specimen and not ourselves. And the second thing here you can see something detector. We're going to talk about what are these and we're going to talk about how are these screens also inserted in these microscope. So let's get started with it. Now we're going to talk about the parts. There's also principle involved in this confocal microscope, but I will talk to you about the principle in the another video because principle if I talk about in the initials it will not be clear and I want you to talk about parts and working before talking to you about the principle. So let's get started. Now the basic parts of the microscope are already present, okay? But what are the different microscope parts that are here? Here are five different microscopes part, okay? First is laser source. Light source is there, but it is the laser source now, which help us to producing monochromatic single wavelength UV light rays of high intensity. First thing is this. Now we gonna have confocal pinholes and excitation filters or beam filters we can say. These are placed after laser source and it is not always important that we gonna get confocal pinholes and excitation filters both inserted into the microscope. It might be possible that they're both present at the same time or it might be possible that only one of them is present and if it's one of them it will be confocal pinhole. So it is not necessary that our excitation filter or beam filter are always present in the microscope. It might or might not be present. But what are their functions? Their function is to produce pointed laser light and they help us to eliminate other unrequired light rays. What do we mean by it? So what's happening in this confocal microscope, guys? Whenever the light is hitting our specimen, we are ensuring that we focus that light ray only at a point and not to other points. Okay, so if we want to focus our light rays only at a point, we need to ensure that all the light rays hitting the other points should get eliminated, and that's the exactly function and that's exactly what our confocal pinholes do. Dichromatic mirror helps to reflect light and allows only fluorescent light rays to pass through. Now, of course, we gonna use not the stained specimen over here, but the fluorescent stained specimen. First thing is this. And the second thing, the function of dichromatic mirror, as I just said, we are focusing onto the point and we don't want light rays coming from other points to hit our detector. We're gonna talk about detector in the next thing. So to ensure that only light rays coming from our point, we are using dichromatic mirror. Now what it do, basically when light rays come through objective lens after hitting the specimen, there are reflected light as well as fluorescent light. So what dichromatic mirror is doing, it is removing reflected light and allowing only fluorescent light to pass through. I hope that's clear. Confocal pinhole before detector. There's another confocal pinhole before detector that help us to eliminate out of focus light rays. These light rays are fluorescent light rays and those fluorescent light rays that are coming out of the focus plane are getting eliminated. Okay, and the other thing is photomultiplier detector. Photomultiplier detector collects all the focus light coming from our spotted point and it helps us to create a 3D image. Now see, we are getting 3D image over here. If the paths are not clear, look at this thing. It'll make your things more clear. But before that, let's talk about what are out focus and what are in focus light rays. Out of focus is simply the light rays coming from non-considered point. Like if we just want to, let's say, want to consider this point. So light rays are also generating from this background and light rays are also generating from this background. Okay. So what are we doing? We are just taking these light rays coming from the spotted point. See here, this is the sample and this is your focal planes. Okay, the thickness, on the basis of a thickness we have divided focal planes. So if I say my focus is just this point, but what is happening, light rays are also coming from this focal plane, this focal plane. So what we mean, we just want this light rays to be considered and we want these light rays to be eliminated. Let's now talk about the parts again, but in the form of picture. Okay, so as I spoke, there's a laser source which help us to getting UV light rays in a monochromatic way. Okay, which is single wavelength. So this is producing light rays and we only want a single wavelength light rays to hit our object, to hit our specimen. So what are we doing? We are using confocal pinholes to eliminate all the unrequired light rays and ensuring that only specific wavelength hits our specimen. Okay, but before hitting a specimen, it is passing through confocal pinholes, it is passing through beam splitter. As I told you, it might or might not be there. Then it is here only, it is not shown in the picture. Here only we gotta get dichromatic mirror. So it passes through dichromatic mirror and then here, as you can see, it passes through dichromatic mirror and then it finally passes through objective and hits our specimen. The process is bit different this time. Okay, so light rays hitting our specimen and out of focus and in focus light rays are generated. Now these out of focus and in focus light rays again pass through objective lens. Then these pass through dichromatic mirror. Here in this dichromatic mirror, all the light rays that are reflected gets eliminated and only fluorescent light that are coming from the specimen reaches the confocal pinholes. Okay, if you see here, then all the light rays that are reflected light rays gets eliminated with the help of dichromatic mirror and only fluorescent light passes through dichromatic mirror and hits our confocal pinholes. So you really need to understand guys, because both the images are not that clear. Here we do not having your confocal pinholes, here we're not having your dichromatic mirror. So it's okay, we can manage, right? So confocal pinholes receives out of focus and in focus fluorescent light rays, and as the function of confocal pinholes, it only allows in focus light rays to pass through it, and finally our detector receives all in focus light rays and helps to get the proper image. But now you can see we are just focusing this point. What about the whole specimen? What we're gonna do, we're gonna change the direction of laser source so it hits our another point of consideration, and then we're gonna receive our image, our light rays from that point, and we're gonna eliminate all other out of focus light rays. And that's how bit by bit our image is formed, by receiving bit by bit light rays. So now finally let's talk about the working of microscope. See, laser source turned on. Here your laser source is turned on. Monochromatic light hit confocal pinhole. But to ensure that only confocal monochromatic light hits our specimen, we uses these confocal pinholes. Okay, so these pinholes ensure that even if there's another kind of wavelength that is coming through laser, will be eliminated before reaching the specimen. So monochromatic light hit confocal pinhole and just take this splitter as dichromatic mirror. Okay, beam splitter is generally not there, but dichromatic mirror is always there, so just take this as dichromatic mirror. Okay, so now these monochromatic light rays hit the specimen via dichromatic mirror and then objective. Okay, um of course, confocal pinhole removes all the unwanted light rays, and then they pass through the dichromatic mirror, and finally our fluorescent stained specimen gets hit, and at a spotted point. Only in focus plane we need to care about. All the out focus planes that are generated needs to be eliminated. So in focus and out focus light rays generated. Now why are these out of focus planes are generated? This is because our if even if we are pointing at a cell, or even if we are pointing at a structure, there must be neighboring areas to it, and light is not only hitting the spot point but all the neighboring areas to it. So we are ensuring that only the light rays coming from that point reaches our objective and finally reaches our detector. So light in focus and out focus light rays generated. Both the light rays hit the dichromatic mirror via objective. All the reflected light coming from the specimen gets eliminated. Fluorescent light only pass through dichromatic mirror, both out of focus and in focus. Again, confocal pinhole removes out of focus plane light rays, and then only in focus fluorescent light reaches our detector. See, so it gets eliminated, in focus light rays reach detector, and finally 3D images form by taking more in focus light rays, because as I said, we are pointing only to a structure, and if we want to get the image of whole specimen, what we will do, we'll change the direction of our laser source, and then we'll focus our point on this structure, and then we'll finally eliminate all those out focus. And if we want to get the specimen structure over here, we'll change the direction again and make sure that the light rays hit now. So what are we doing? We are bit by bit collecting the details of the specimen, and then using computer we are making the 3D final image. Now this is again the repetition, and if you have still doubts, please comment them in the comment section. I love to answer them all. All the principle and advantages and the images formed by the confocal microscope will be covered in the next video. Till then, take care and bye bye.